Hello. Hello, everyone. I hope the sound is clear. Today, actually, I was not able to just give my full presentation view. So, from tomorrow, from the next lecture, I will show this entire lectures in the same format in which we were earlier conducting. Right, I will be here on this full page, or we can say full display of my entire image, so that it will be more presentable and you can have more attentions to also. Because what the case is with this, whenever I am just showing it in one of the corners, so many of the students are not able to give that much attention. So from tomorrow we will just shift to the our earlier editions of our lecture. Okay. So I will start in within 2 minutes. We will just wait for the 2 minutes more. Uh, okay. So before that, I will give you an idea that today our the pending lectures of cell biology are going to be conducted in the next 10 days, and the whatever the delay that was been made, that was because as you know, I have already started the paid batches from the 6th of November. So it was quite difficult for me to arrange the both of the batches at the one time. So what I have decided is that from this or from tomorrow, I am going to conduct the same lectures even in the paid batch also, and even in the free YouTube batches also. Obviously, the content of these free YouTube lectures would be the lesser than these paid batches. The detailed notes will be there in the paid batch. So this is the case for this rest of the part of the cell biology. On in this, then our next module, that is the physiology module. In the case of that physiology module also, I am going to conduct the lectures in parallel with this paid batch and the live free batch also. The same topic will be taught in the paid batch also, and the same would be in this live YouTube lectures also. And it will start around this 1st of December, because there are some schedules delays are there which are obvious, because in these last days I was also involved in making those previous year question booklets of this IF Indian Forest Service also, and CSE, and just combining both of them. So it took a lot of time also in this cell biology module at the same time. So we'll start it around 1st of December. The admissions for that session will be launched soon. So yes, let's start with this today's lecture. Before that, if you have not joined this cell biology module, and if you want to join, you can have the at least recorded lecture of this all of this cell module. There are already 57 students are who have already joined, and most likely they will also join this physiology module also. So you can ask for that. So let's start with this our today's lecture. That is about the meiosis. Okay, we have seen this mitosis, we have seen this cell cycle regulation also in the along with the interphase also. So what we are going to see today, right now, is, see, if you have any query, you can also ask at the same time. Okay, I will attend your query on this while this live chat on. Okay. I can see this. There are 9 students are there. So if any one of you have any doubt regarding this lecture, you can ask that doubt in the chat box. I will attend those doubts. Okay. So let's start. See, the meiosis is nothing but earlier we have seen this mitosis where this diploid cell was giving rise to formation of diploid cell only. Okay, two daughter cells only. But in the case of meiosis, what is happening is that whatever these daughter cells are forming, there were 4 haploid cells will form. Okay, so that is the case in the case of this meiosis, that the haploid cells are forming in this. So let's see. And as the number of chromosomes are decreasing, this diploid cell is becoming this haploid cell. So that's why this is also called as what, reductive cell division. Okay. Now there are multiple phases in this meiosis. We are going to see, but this is the general diagram of this meiosis is. Okay. This is what happens in the mitosis. This diploid cells gives rise to the formation of 2 diploid cells. But in the case of this meiosis, you can see this is the DNA replication phase, as it was in the case of mitosis. Here also the DNA replication phase would be there, and this DNA replication phase, after this DNA replication, it would follow by meiosis I and meiosis II. What will happen in this meiosis I is this number of chromosomes. You can see here is the 2n, means diploid cell is converting into the haploid cell. So this is why this meiosis I is called as what? Reductional division. Okay. The next thing is meiosis II, in which these two cells are giving rise to the formation of two haploid cells each. Means total four haploid cells will be formed. And that, as the number of chromosomes are not further halved, they are just remain as n to n, means there is no reduction in the number of chromosomes. It is called as, this meiosis II is called as what? Equational division. Okay. Now I hope you have just understood this meiosis I and meiosis II. Now we are going to see these are the different phases which we are going to study. This is the first part before this meiosis I or prophase, meiotic prophase, that is this preparation of meiosis, the S phase, in which this DNA is going to be duplicated. Then there will be a second phase or the second part of this entire meiosis, that is the meiotic prophase. And there are different phases as compared to the prophase of mitosis. This prophase of meiosis is very different. So this meiotic prophase is having these multiple stages or multiple phases like this: leptotene, zygotene, pachytene, diplotene, diakinesis. Okay, this bouquet stage is one of, considered as one of the substage of this leptotene stage. Okay, so let's see now this in the last part of this meiosis one. What are the other phases? That is this metaphase one, anaphase one, and telophase one. After the telophase one, the cytokinesis will occur, and we will form these two cells, and these two cells will again form this two haploid cell each. So this is the entire diagram of this meiosis. Okay. Now let's see what happens in this preparation of meiosis. So this is the cell which is about to undergo the first phase of this meiosis. What happened in this is that whatever these chromosomes are there inside. Inside, you must know that there are 23 pairs of chromosomes are present. Out of these 23 pairs, one pair is the sex chromosome, that is either XS in case of females and XY in case of males. So whatever these pairs of chromosomes are there, 22 pairs, which are called as what? Autosomal, because they just determine everything else other than the sex. And this one pair, 23rd pair, is called as sex chromosome. Okay. So whatever these autosomal sex chromosomes are, or autosomal DNA pairs, chromosomal pairs are, they are present as an homologous chromosome in between. Means you can see here, this is an homologous pair of chromosome. Similar, you can say, having almost same function. So it is, this is the one pair, means two chromosome. This is again another homologous pair. You can see this is looking like somewhat similar. So that's why it is called, this is the second pair of chromosome. Okay. So such 22 pairs of chromosomes will be there. But as for our simplicity, we cannot draw all of them. I have just shown here only two homologous pairs of chromosomes. So let's see what happened in case of those two homologous pairs, so that you will understand everything that is happening in this meiosis. So obviously they will undergo, before starting this prophase, they will undergo this, or before starting this meiosis one, they will undergo the pre-meiotic S phase. And as in the case of mitosis, this S phase, this DNA is going to be duplicated. So you can see this was the first strand of DNA. Now it has another strand with it. So this is the duplicated homologous chromosome. This another second chromosome was similar like this, but this has also again duplicated. Right. Same case with this also. Earlier there were two homologous chromosomes, but in the S phase both of them have been duplicated. Okay. So this is how, this is what occurs in this S phase, and these duplicated chromosomes will be fused at a point that is referred to as centromere. Okay. Now let's see the first. We are going for the going to see the prophase of this meiosis one. In that prophase, there are multiple phases. Like the first step is what? Leptotene. The sub-stage of this is called as Bouquet stage. Second one which is Zygotene. Okay. And later on, this will be the Pachytene, Diplotene, and diakinesis. Okay. So let's start with this Leptotene stage. Now, what is happening in this Leptotene stage is that whatever these earlier duplicated chromosomes are there, at their end there is a presence of repetitive DNA sequences. Okay, this is the fixed characteristic of DNA structure. Many of you might not know this, but we will study this in the chromosome structure. Okay, so at the suppose this is the that X-shaped chromosome. What is the characteristic of this X chromosome? Is it at the end there are DNA sequences, nucleotide sequences there are, which are called as what? Telomeres. Telomeres. So these chromosomes will also have telomeres like this. You can see there are three telomeres are here, and those telomeres attaches this entire chromosomes to where? To this nuclear membrane. You can see most of these chromosomes are now attached to this DNA, okay, to this nuclear membrane. So that attachment will be with the help of what? With the help of this telomeric region of this chromosome. So right now we can see they have been attached. But what happened in the next phase is that these chromosomes and their telomeres, telomeric region of these chromosomes travels along this entire pathway of nuclear membrane. This will travel here, this will travel here, this will travel here, this will travel here. And in some way, at some region, all of these telomeric regions will aggregate like this. And when they aggregate at some region of this nuclear membrane, they looks like a bouquet of flowers. You can see this is looking like a bouquet of flowers. So that's why this stage is referred to as what? Bouquet stage. Now let's see what happens in this zygotene stage. In the zygotene, see the significance of this bouquet stage is that all of the homologous, even the homologous chromosomes which were quite apart from each other. Here you can see this was the homologous chromosome and this was the second homologous chromosome here. So they were quite apart from each other. But in the after this bouquet state, most of them get in close proximity, so that can align themselves side by side. Suppose this is the one strand of this homologous chromosome, duplicated strand, and this is the another duplicated chromosome, homologous chromosome. Okay. As in the case of bouquet stage, most of their telomeres have just aggregated at one point, so they come in close proximity. Now, once they come in close proximity, they can align themselves side by side. So they will form a structure like this, a structure which is in a close proximity to each other. At the same time, there would be initiation of synapsis. Now, what is this synapsis? Synapsis is nothing but the central elemental proteins. There will be some proteins will be here, which are holding these two chromosomes together. Okay, got it? What this synapsis or the synaptonemal complex is? What this synaptonemal complex is doing? These are just nothing but holding these two two strands of DNA together. That the initiation of that formation of that complex will start in zygotene stage only. Here you can see these both chromosomes have came in close contact with each other. They have aligned themselves along the side by side or in close proximity. The next thing that is the Pachytene, what will happen is that whatever this chromosome that have come in close proximity to each other, now they will form a recombination nodules. Means what? A part of their DNA strands will form a cross, and at that cross, after that crossing, there will be the recombination, there will be the exchange of DNA fragments would be there. So that exchange of DNA fragments is called as what? Homologous recombination. Okay, and this entire protein complex which is holding these two chromosomes together, it is called a synaptonemal complex. Okay, so that's why these are called as recombination modules, and they occur in which stage? Pachytene stage. The next is Diplotene stage. And what happens in this Diplotene stage? See, after this recombination, what will happen is that part of DNA fragment from this strand will go to this strand, and part of DNA fragments from this strand will go to this strand. Okay, this strand to this strand. So there would be an exchange of DNA fragments in between two strands. So after that exchange, what is the structure is going to be formed? That is called as what? This chiasmata. Chiasmata is nothing but what? Chiasmata is nothing but these duplicated chromosomes which are formed after this recombination. Okay, and whatever the earlier synaptonemal complex was there, which was actually holding these two chromosomes together, now disassembles. Now there is no more synaptonemal complex is there. Now they are hold together only at this chiasmata point, okay, or only at the point of this crossover. You can see. Otherwise, other part of this you can see here, they are just hold together by throughout their entire length because of the synaptonemal complex. But in this case, they are just hold together at this chiasmata only, because other synaptonemal complex at this point has been dismantled. Now let's see what happens in the diakinesis. See, in the diakinesis, the important phase that is occurring is this: the nuclear membrane will disrupt. Okay. There is no nuclear membrane in this diakinesis. And the next phase that is going to be occurring is that whatever this DNA is there, you can see the DNA in this phase is what? Condensed as compared to this. So the DNA condensation is also done in this. So Deepika is asking telomeres. Okay. Chiasmata is crossing over. Yes. Telomeres are just the region present which are at the end part of this chromosome. They are called as telomeres. And chiasmata means what? They are present, formed at this crossover DNA. Right here, this region is called as chiasmata. Okay. Now the next phase is what? We have just completed prophase of meiosis I. Now we are going to conduct metaphase of meiosis I. So that's why we called as metaphase 1. In the same case, anaphase, telophase are called as anaphase 1, telophase 1. So what is going to be happen in the metaphase 1? Whatever these two strands of chromosomes are there, now this chromosome will go to this pole, and this chromosome homologous pair will go to this chromosome. Then this will look like this. Okay. Same case with this also. Half of the homologous chromosomes will travel to this pole, and another half will travel to this pole. That is, so this alignment of this entire chromosomes, homologous chromosomes at this equator will occur at this metaphase 1. In the anaphase, now the anaphase 1, this whatever this chiasmata was there, earlier formed chiasmata was there. Now that chiasmata will be broke apart, completely disassembled. So after that complete disassembly, these chromosomes are now free. They can travel to the respective poles, and can see in the anaphase 1 they have traveled. In the telophase 1, these chromosomes will reach to their respective centrosome, and this nuclear membrane will be reformed. Once this nuclear membrane is reformed, the cell is ready to go for cytokinesis. So there will be the cytokinesis one, after which these two cells will be formed, 1 and 2. Okay. Now they will be haploid because DNA has already been halved within this separation. So that's why the whatever these progenies are there, they will be also haploid, because this rest of the transition is there. That is same as the in case of the mitosis. Okay. This is also the same as that in case of this mitosis. Okay. So that's why these haploid cells are giving two haploid cells as in the case of mitosis, and this haploid cell will give this two haploid cell. Okay. The processes will be almost same like the mitosis. Okay. You will have to replace the diploid cell with the haploid cell. So that is what happening in this meiosis, and right now I will just give you a quick an animation type tour of this entire meiosis. And see, this is the actually diagram of whatever these 23 pairs of chromosomes we have seen. These are the 22 different types of chromosomes. Okay, obviously they are not the same. Okay, these are just for to represent, or just to make you sure that these are the things which are present in there. So such different structures of chromosomes would be there, and the 23rd pair is XS in the case of females and XY in the case of males. So we will study out of this only two. So these are the two present inside this entire meiotic cell. So this is the one strand, this is the another homologous strand. Now what will happen in the next phase? You can see this gets duplicated, and this duplicated stage is called as what? Pre-meiotic S phase. Now what is happening in the leptotene phase? You can see these telomeres are getting attached with this nuclear membrane. The next part, that is with the help of another protein that is called as a link protein. Okay, you can mention this protein also in addition to this telomeres. So in the next phase, what is happening is that they are this. You can see these telomeres are traveling along this entire mitochondria and entire membrane, nuclear membrane. Okay, and finally they are forming this bouquet-like stage, right? So this is referred to as bouquet stage. The next phase is once they have come in close proximity, now they can a part of their these telomeres will get released from this nuclear membrane, and they can align side by side along with each other. And after, and the same also in the same phase only, in the zygotene phase only, the synaptonemal complex, here you can see this synaptonemal complex has been formed, right? Whatever this part is you are seeing, this is the synaptonemal complex. Okay. So after that, the next phase will arise, that is the Pachytene, in which this crossover will occur. That is referred to as what? Recombination nodule, because the recombination occurred that point. The next phase is these are the different proteins that are acting in this recombination. You can just mention these names of the protein. After this recombination, these proteins will form a chiasmata. This is the chiasmata, a cross structure formed after the crossover. Once the chiasmata is formed in the diplotene stage, in the diakinesis phase, they will just condense. This structure of DNA is just condensed in this phase. After that condensation, they align themselves at this metaphase plate or metaphase 1. After that metaphase plate alignment, they will get segregated into the respective poles. So after that segregation, the new nuclear membrane will form around it themselves in telophase 1. After the nuclear membrane formation, the cell is ready to go for the cytokinesis. So these will the cells will be formed, and ultimately we are getting these two haploid cell from this cell and two haploid cell from this cell. So these are the four haploid cell from one, you can say, diploid cell. So this is what the entire meiosis is. Okay. And if you have any query, you can ask. कुछ query है क्या? Do you have any query? If you have any query, you can ask. Otherwise, we will stop this live session here. We will conduct the next live session tomorrow at 5 p.m. only. Okay. I will announce which chapter we are going to study prior to the lecture. In the morning only, I will announce the next chapter that we are going to study. And I will just also upload this lecture immediately on YouTube, so you can watch this recorded video of this part also. Okay. Okay. So there are no queries. So we will stop here. Tomorrow we will meet for the next part of this free lecture series. So till then, enjoy. Have a nice day. Good night.